Hello everyone. Uh, we are now going to start with our next lecture, that is various structural level of proteins. I am Himanshu Jaiswal, Assistant Professor, Department of Botany, J.D. Patil Sanglodkar Mahavidyalaya, Daryapur. Friends, there are different structural level of proteins such as primary structure, secondary structure, super secondary structure, tertiary structure and quaternary structure. The primary structure of protein is just a sequence of amino acids which is mainly formed or stabilized by the peptide bonds. It ranges from the NH3 terminus to the COO terminus. The primary structure of the protein is depending on the sequence of codons present in the mRNA. This codons will determine how the amino acids will be placed, and finally the primary structure of protein. This primary structure of protein will ultimately decide any higher level structure of protein, that is secondary structure, super secondary structure, tertiary structure and quaternary structure. Now the secondary structure of protein is formed by local folding of a polypeptide upon itself, right? So it is formed by local folding of polypeptide upon itself. Once a polypeptide folds upon itself, then there is formation of a secondary structure. This structure is mainly stabilized by the hydrogen bonds, which are a type of weak interactions. There are three most common secondary structures: alpha helix, beta sheets and beta turns. Alpha helix is a coiled structure of protein which is generally stabilized by the hydrogen bond. There is due to the coiling, the distance between two amino acids becomes 1.5 Armstrong, which is actually 3.8 Armstrong in the straight chain of protein. There are 3.6 amino acids present per helical turn. So every amino acid is rotated by 100 degrees. So 3.6 amino acids are going to give you a rotation of 360 degrees. So one helical turn is composed of 3.6 amino acids. The hydrogen bonds are formed between N and N plus 4th amino acid, that is first and fifth amino acid are engaged in the hydrogen bonding. So first amino acid is associated with fifth amino acid, then fifth will be associated with the 10th amino acid and so on. Now one of the example of this alpha helix containing protein is aquaporine. Aquaporines are water channels present on the membrane of mammalian cells. These water channels are made up of 4 subunits or 4 identical subunits which are having 28 kilodalton molecular weight, each subunit of which is composed of 6 membrane spanning alpha helices. Now another is beta sheet. Beta sheet is formed by side alignment of 2 zigzag shaped polypeptides. The distance between two amino acids in a fully extended beta sheet is 3.5 Armstrong degrees. There are two types of beta sheets. One is parallel beta sheet and another is anti-parallel beta sheets. The anti-parallel beta sheets are quite stable as compared to the parallel beta sheets due to the linear hydrogen bonding present in the anti-parallel beta sheets. Now one of the example of this beta sheet containing structure is a porine protein which is present on the mitochondrial outer membrane. These porines are trimers which are composed of 3 identical subunits, each subunit of which is composed of 16 antiparallel beta strands which form a barrel shaped structure in the protein, and that allows the molecules to pass through it. Now the super secondary structures are formed by a combination of 2 or more secondary structures. Generally they are called as motifs. So there are various types of motifs such as DNA binding motif, zinc finger motif, leucine zipper motif. So they are super secondary structures, means they contain 2 or more secondary structures. The next and important type of structure is a tertiary structure. It is a 3-dimensional structure of protein. Right? The tertiary structure is formed by when you place a protein in the medium of water, the water will repel hydrophobic amino acids to the core of protein, and hydrophobic amino acids will come in contact with water. There is favorable interaction with the hydrophilic amino acids and water, and due to this there is formation of a tertiary structure of protein. So tertiary structure is a three-dimensional structure of protein. Now the next structural level is quaternary structure. Quaternary structure is formed by combination of two or more tertiary structures. Means when two or more tertiary structured proteins combine with each other, there is formation of a quaternary structure. These tertiary structures are associated with each other with the help of various types of non-covalent interactions such as covalent bonds, hydrogen bonding, electrostatic interaction, hydrophobic interaction, and van der Waals interaction. Thank you so much for patiently listening to me.